Good afternoon, and I welcome you to the talk on how to isolate stem cells. So this talk will deal with different types of stem cells and the isolation of culture, and we will be focusing mostly on mesenchymal stem cells and cancer stem cells. So but first I'll introduce to a few key concepts. Who knows what is a stem cell?
Stem cell can differently can self-renew.
Very correct. So a cancer stem cell or any kind of stem cell has the capability to divide and form more of its own, which is called self-renewal. So it renews its population, as well as it differentiates into different cell types. So cancer stem cells or any mesenchymal stem cells or any stem cell that you know about, they are all unspecialized, which means they are undifferentiated. They are capable of dividing and renewing themselves, which is for a prolonged time, and they have the potential to give rise to specialized tissues, which is known as differentiation. So what is differentiation? It is the process of forming specialized tissues, and once the differentiation has occurred, a differentiated cell usually is not able to go back to its progenitor type or embryonic stem cell type. For example, if a embryonic stem cell differentiates and form a liver cell, the liver cell cannot go back and become an embryonic stem cell. Neither can the liver cell become another differentiated type of cell. For example, it cannot become a nerve cell. Once it's differentiated, the cell type is specific for that type. There are a few stem cell terminologies that you should be aware of. Potency is the measure of how many types of stem cells, how many types of specialized cells a stem cell can give rise to. There are 3 kinds of potency. A stem cell can be totipotent, pluripotent, or multipotent. A totipotent stem cell has the potential to develop into a new individual. For example, when you have a fertilized egg and it splits and forms 2 cells, each of those cells have the potential to give rise to a new individual. That's how you get zygotic twins. The next is pluripotent. This type of stem cell has the potential to give rise to all the specialized cells that's present in your body. For example, embryonic stem cell. An embryonic stem cell can differentiate and form every tissue that's present in your body. The next type is multipotent. This can give rise to multiple types of specialized cells, but not all types. For example, tissue stem cells. Stem cells are maintained by a network of genes. The three most important genes regulating stem cells are NANOG, PO5F1, also known as OCT4, and SOX2. These 3 are very critical in maintaining stemness. These interact with other genes, for example, MYC or STAT3, and this is how stemness is maintained. Stemness can be regulated by 4 different pathways: WNT signaling, Notch signaling, P10 signaling, Hedgehog signaling. I will not go into too much of details into this because there is a lack of time, and we'll focus more on the isolating of stem cells. So there are basically, if you consider where they are found, stem cells are classified into 4 main types: embryonic stem cells, induced pluripotent stem cells, tissue stem cells, and cancer stem cells. So first we'll talk about embryonic stem cells. So when a fertilized egg divides, it forms into a mass of cells called blastocyst. And in that, there is this inner mass of cells. This is where the embryonic stem cells are present. So this is a microscopic image of a blastocyst, and this is where the embryonic stem cells are located. So how would you isolate embryonic stem cells? So you can isolate embryonic stem cells from donated excess in vitro fertilized embryos. You have the fertilized egg, which divides and forms a blastocyst, and then from the inner cell mass, you get the embryonic stem cells. Another way you can have embryonic stem cells is by cloning. You can have a donated egg, a donated cell. You can fuse them, and this forms a new embryo. When this embryo divides and forms a blastocyst, from the inner mass, you can isolate the stem cells. So here's the blastocyst, winner mass and you harvest that, and you can culture them in your lab and have embryonic stem cells growing in your lab. To culture embryonic stem cells, you would need a platform to grow them. So you can have a plastic culture dish, but usually what is happening, or what scientists usually do, is that they coat it with a feeder layer. This is usually comprised of mouse embryonic fibroblasts. So these cells are called feeder layer because it feeds the cancer stem cells or any stem cell that you're growing. Usually you're using embryonic stem cells in this case. So you would coat them, and these cells will provide nutrition and growth factors so that the embryonic stem cells would proliferate. You can also grow them without a feeder-free, in a feeder-free condition, but then you would have to supplement the growth factors in the culture media. And when you have enough cells and they are crowded, you can divide them and grow them and expand them. So this is how embryonic stem cells look in a culture dish. So the central spheroid is the embryonic stem cells, and the background, you see the spindle-shaped cells. Those are the mouse embryonic fibroblasts or the feeder layer. So when you have a lot of embryonic stem cells, you can divide them and you can have different colonies or embryonic bodies. So an embryonic stem cell is pluripotent, and under proper conditions, it can give rise to skin. In another specific condition, it can give rise to neurons. In another specific condition, it can give rise to blood or liver or whatever you want. It has the potential, it's pluripotent, it has the potential to form any tissue that you want in a human body. Yes?
How could you collect stem cells from the feeder cells?
Feeder cells?
Yeah,
Trypsinization.
Oh, okay.
You have to trypsinize them. Trypsinize them and get them, and to make
So
sure. Yes.
Sorry. So in the next passage, all of the cells they are mixed together.
So here's the thing. It's a little tricky to get the embryonic stem cell because, well, there are two layers. The first layer that comes off is obviously the embryonic stem cells. If your trypsin is too much, then the mouse feeder layer will come up. To make sure that what you're getting is embryonic stem cell is to characterize it whenever you're doing an experiment. So you'll have to look for embryonic stem cell markers. So the next type that I'm going to talk about is induced pluripotent stem cells. So induced pluripotent stem cells can be obtained by taking a cell from a donor and reprogramming it genetically so that it starts to behave like pluripotent stem cell or an embryonic stem cell. So you are inducing a cell to behave like a stem cell. So by this, you can differentiate them and form any different type of tissue that you would like. For example, if you have induced pluripotent stem cells growing in your lab, you can differentiate into any type of cells, for example, cartilage, bone, neural tissue, etc. And the main advantage of this is you don't need embryos. So usually it's done in clinical stem cell research. You take cells from a patient, usually skin or fibroblast. You treat them with reprogramming factors. You wait for them to become pluripotent, and then you generate the type of tissue that the patient needs. For example, if this patient has a skin injury or a burn, you can make them into a skin and then do surgery on the patient and help them out. So the third type of stem cell is tissue or adult stem cell. It's more proper to call them tissue stem cells than to call them adult stem cells, but it's also known as adult stem cells. So you can find them in the brain, in the dental pulp, in bone marrow. So almost all tissues have tissue stem cells. They are mostly responsible for repair and repairing your damaged tissue. So what can these tissue stem cells do? So tissue stem cells are multipotent. They cannot give rise to all the tissues in your body. They have a specific set of tissues or cells that they can give rise to. For example, a blood stem cell, which is also known as hematopoietic stem cell. It's found in bone marrow, and it can differentiate and give rise to different components of your blood. For example, macrophages or platelets, etc. So these are multipotent. Another tissue stem cell is a neural stem cell. It can give rise to different cell types, for example, neurons, oligodendrocytes, astrocytes, etc. Gut stem cells can give rise to goblet cells or endocrine cells, any cell that's in your intestine. Hematopoietic stem cells or the blood stem cells, they can give rise to a whole set of cells, for example, T cell, B cell, dendritic cell, etc. Mesenchymal stem cells are the stem cells that are capable of giving rise to bones, cartilage, and fat. So isolation of mesenchymal stem cells. You can isolate mesenchymal stem cells from bone marrow, from adipose tissue, and cord blood. So now we'll talk about how to isolate mesenchymal stem cells from the bone marrow. So bone marrow is found in spongy bone, and these bone marrow stem cells, they give rise to different cells like osteoblasts, which form the bone, chondrocytes, which form the cartilage, myoblasts, which form the muscles, etc., and so on. So these are the materials that we would require for harvesting and culturing mesenchymal stem cells. Basically, you need a harvest buffer to harvest the mesenchymal stem cells, and you would need a culture media to culture the mesenchymal stem cells. And you would need some equipment for culturing them in a sterile condition, as well as an incubator to grow them in proper control conditions. So the following images have some graphic content on animal dissection and tumor. If you feel offended, please, you may want to take a moment. So this is, now we are going to talk about how to isolate mouse mesenchymal stem cells. So you have a mice which is clean and euthanized, and then you would cut out this outer skin layer. Then slowly would start cutting in deeper, and then cut out the whole leg portion. After you have done that, you would cut off the paws, because the paws are mostly formed of cartilage, and we are not interested in that. So then you put it in a media to keep it moist so that it doesn't dry out. You perform the same thing on the next leg, and then you have your two flanks. Then you dissect the portion to remove all the connective tissue, and you have to really be careful to really clean and get just the bone, because if you have any other cell, it will contaminate what you want. So after you're done that, you're done cleaning the bone, you can put the bone piece into a harvest media. After you're done putting them in the harvest media, you take a 27 gauge needle syringe and flush out the bone marrow. You have to do this really forcefully so that you can get as much as bone marrow as possible. After you are done getting all the bone marrow you could get, you let them attach them for 2 days. After 2 days, you remove the media. There might be floating cells, which are mostly blood cells. We do not want them. We want the attached cells, and from there you can pick colonies and grow them and expand them. Very important, after you're done having enough cells, you have to characterize them. You want to make sure that what you have is mouse mesenchymal stem cell. So you look for mouse mesenchymal stem cell marker, for example, CD44, CD90, SCA1, etc. It's very similar for what you would do to isolate mesenchymal stems from a human. You would isolate bone marrow from a human, and here you would look for human mesenchymal stem cell markers. For example, endoglin, CD44, etc. You can isolate mesenchymal stem cells from adipose tissue. This is very helpful. For example, a patient who's having liposuction surgery, he doesn't need the liposuction, the tissue left out after liposuction. From there, you can actually isolate mesenchymal stem cells. So you take the fatty tissue, digest it with collagenase, and then you get a pellet. And you can grow this pellet in a culture condition, characterize them for mesenchymal tissue markers, mesenchymal stem cell markers, and then you can differentiate them into tissue for whatever the patient might need. You can isolate mesenchymal stem cells from cord blood. So you harvest cord blood, You mix it in a 3:1 ratio with RPMI media. You use a fecal park medium, which is a density gradient medium. It helps you to divide or like separate out layers in your media on the basis of different densities. For example, when you put your harvested blood, cord blood, with fecal park medium, you get a floating intersection of cells, which are the mesenchymal stem cells. It is between the fecal park medium and the plasma. So you isolate that, centrifuge it, and you again get a pellet. And you harvest that and grow them, and again characterize them for mesenchymal stem cell marker. Make sure what you have is actually mesenchymal stem cell, not any other contaminant cell type. You can take these mesenchymal stem cells, put them in an adipogenic media, get adipocytes or fatty tissue. You can culture them in osteogenic differentiation media, get osteocytes and form bones. You can put them in chondrogenic differentiation media, culture them, get chondrocytes, and get cartilage. The fourth stem cell type that we were discussing is a cancer stem cell. So a basic cancer stem niche consists of a cancer stem cell and a non-stem cancer cell. It's a cancer cell, does not have stem properties. So cancer stem cell, like any other stem cell, can divide and self-renew, form more of its own, as well as differentiate to form a non-stem cancer cell. However, a non-stem cancer cell divides to form only more of its own type, which is a non-stem cancer cell. It cannot form a cancer stem cell usually. There are different cancer stem markers for different cancer types. For example, for a human brain cancer stem cell, the markers are CD133. For a mammary gland or breast cancer stem type, you would have CD44 positive, CD130 positive, etc. For prostate, it's again different. There are some integrins that are highly expressed in them, and they have SCA1 expression. So for this, you take any tumor or any cancerous tissue that you want to isolate cancer stem cells from. You chop them into smaller pieces, enzymatically digest them using collagenase, use a 70 micron sieve to divide the clumps. You culture them in proper culture media, and at the last but not the least, you have to sort them cytometrically. So these are the steps, and then you cytometrically sort them using the cell surface markers specific for the cancer stem type that you're looking for. So here we'll maybe consider CD44 and CD133 for prostate cancer. So for example, you take this single cell suspension, you mark them with your required markers, then you sort them using a flow cytometer into positive population and negative population. So the positive population is the one that you're interested in, the cancer stem cells, and the negative population is the non-stem cancer population. And then you look for expression of stem genes to confirm that what you have is a cancer stem cell. There is another way of isolating cancer stem cells. It's called magnetic activated cell sorting. It's actually very similar to what we just discussed. Here we label the cancer stem cells with primary antibodies, and we label the secondary antibodies are conjugated to a magnetic bead. So after we mix them, we use a magnet to pellet the cancer stem cells down. Then we characterize them again for the cell surface marker, as well as for expression of their stemness genes, for example NANOG. You can do that by Western blot or by RT-PCR. So another thing to really keep in mind is using growth factors for the cancer stem type that you're using. For example, if you're growing estrogen dependent breast cancer, you have to supplement your media with estrogen. If you're using androgen dependent prostate cancer, you have to keep in mind that you have to supplement androgen in your media, and this is specific for whatever cancer type you are using. Then you culture them and expand them. You can grow them as tumor spheres where they form spheroids in a free floating condition, or they can grow them attached where they can spread and grow depending on your culture preference. And then you can study their tumorigenicity, and you can test them for drugs. So summarizing, there are 4 main different types of stem cells: embryonic stem cells, induced pluripotent stem cells, tissue stem cells, for example, mesenchymal stem cells, neural stem cells, cancer stem cells, for example, brain cancer, prostate cancer, etc. The isolation procedure depends on the type of stem cell you're isolating. For example, if you're isolating embryonic stem cells, you would isolate them from the inner mass of the blastocyst. If you want induced pluripotent stem cells, you'll have to induce pluripotency by genetic reprogramming. If you are working on tissue stem cells, you need to isolate them from your tissue of interest. For example, your mesenchymal stem cells, you can isolate them from cord blood or adipose tissue or bone marrow. Cancer stem cells can be isolated from the cancer tissue or the cell line that you're interested in. Characterization of stem cells is very critical. You have to make sure that the cell type you have isolated is actually of what you desire and is not contaminated with any other cell type. So different stem cells have different cell surface markers. Depending on the stem cell type you are interested in, you would like to look for those markers on those cells, as well as you have to see whether your stem cells express the stem genes. And the culture of stem cells is dependent on the type of stem cell that you're growing. For example, an embryonic stem cell would need a feeder layer, or you would supplement nutrition for those in the media, and different cancer types need different growth factors. Thank you for listening. Do you have any questions? Yes. Okay.
When you're isolating the cells from the mesenchymal, from the bone of the mice, how much of the time you have for doing that from the moment you kill the?
It depends on how comfortable you are, but it takes, for me, it takes around 2 hours. But it depends how comfortable you are, and you have to be really careful with the cleaning. The first time you do it, it's really, really laborious.
And it's safe, like for the product of cells that we're going to get, they're still alive. It's a good amount of time for them to be.
That's why you keep them moist. You do not let them dry out. Whenever you're isolating, see, when you take the leg out, you keep it in the media. Don't let the cells dry out. Drying out will lead to death of cells. Obviously, you will lose some cells during the process, but you'll still get a good amount.
And for the stem cells, can we freeze the cells in the?
You can freeze the cells, but I would not recommend it.
Okay. Why would you not recommend it?
Because they are very gentle. You have to be very careful while growing them. They have to have their media changed every 2 days. You have to really monitor them. And when you are freezing them, they actually grow through some harsh conditions, which actually end up killing them or differentiating them. So it is possible. I'm not saying it's completely impossible. You may freeze them and actually be able to get some, but it usually doesn't work out in a good amount.
And how to make passage of how long you can culture?
Right.
So the ones that you can isolate can be passaged till maximum of 3, the primary ones.
Okay.
Even there are some companies who sell stem cell lines, even they would not suggest you passaging more than 4. Forget 4, even the third one, they'll start like not growing and dying. Even though they are known for prolonged self-renewal in the culture conditions in your lab, they are not really suited for growth there.
Any other questions? This is an introductory talk, so don't be shy about asking questions of Dr. Talukdar. Yes.
Yeah. The IPS, the induced pluripotent stem cells, how long does it take for them to grow? I mean, I guess it depends on.
Okay.
I have not really worked with induced pluripotent stem cells. But when you say how long it takes them to grow, what do you mean? How many cells do you want to have for your study? It depends on the number of cells you want, really. So they grow, for example, say, divide and say once 24 hours. So if you have in the beginning a lot of cells, that will help in getting more cells in the end. So number of cells that you start culturing is also important. But they are slow growing. Any primary cell type is slow growing, especially stem cells.
Are you transferring this to T75s or something larger?
Yes. It depends on how many cells you want. So initially, if you have lesser number of cells, you would culture them in a smaller flask, say T25. The more you have, the more you can grow and harvest. It depends on how much cells you need. Like when I'm growing cancer stem cells and I need them for sorting, I grow them in T175, and that too like 4 big ones, because I need really a lot of cells for injecting in mice. So it depends on that. Mesenchymal stems are multipotent, right? So they can form multiple lineages, but not all. Something that's totipotent or pluripotent can give rise to another cell, another stem type or progenitors. But something that's multipotent is kind of limited. So mesenchymal stem cell cannot give rise to an embryonic stem cell. But an embryonic stem cell can give rise to different tissues, and those tissues will have their tissue stem cells, including mesenchymal stem cell.
And did you cover how long it took you to prepare the cells before you could use them? Is it 2 days before?
Which one?
So you're looking at the mesenchymal cells.
Okay.
It takes you, 2 days before you need to do the experiment?
Definitely.
That's when...
Yeah, you need to autoclave your equipment, make sure you have proper culture conditions, culture media, your growth factors, your meiosis, you have it for your dissection. And then, depending on how many cells you need, you'll have to be more careful about how many cells you harvest. If you need just a few, just flush once or twice, you're good. But if you need a lot of cells, you'll probably need a lot of mice as well.
So how do you manipulate the genes, like the transfection, overexpression, or knockdown? So for the transfection or like infection, what's the efficiency?
The efficiencies for stem cells is actually low. So you'll have to work on it and optimize for your cell type. I work with cancer stem cells right now, and initially I was not able to transfect them at all. But I'm using a higher dose of the transfecting agent and an equal amount of the DNA or RNA that you want, and I keep them overnight in a shaking condition, and that seems to be working for now.
Oh, in a shaking condition. Okay.
But it depends. Your cell type might be lesser sensitive or more sensitive. It depends.
Okay. So have you ever tried the virus?
Yes, definitely. Virus definitely has much more efficiency than, DNA transfection or RNA transfection. Virus has way more efficiency. I've used virus. Actually, I use more of virus.
Okay. So what kind of transfection reagent do you use?
I actually use lipofectamine. I know it's not the most efficient, but it seems to work in my case. And if you want to use viruses, lentiviruses are the best for infection.
Any other questions? Okay, well, there's snacks and coffee, and I'd encourage you all to talk with each other, because obviously we're all interested in stem cells, and everyone here is from very different departments. So please meet your neighbors and hang out here for the next half hour if you want to. Thanks for coming. Oh, yeah. Thank you.